All right, so now let's talk about reabsorption. So reabsorption occurs or starts at the proximal convoluted tubule. Most of it occurs in the proximal convoluted tubule. So if you remember, we have our Bowman's capsule. Capsule here, and then over here we have the proximal convoluted tubule. And we've had filtration, so everything's gone into Bowman's capsule. And keep in mind, what gets filtrated into the capsule? Well, everything except red blood cells and or blood cells and proteins. Okay, that means everything else has been filtered out of the blood and is in Bowman's capsule. So that's, ions, potassium, sodium, water, bicarbonate, amino acids, glucose, everything is in there. Okay, so it's all, and that's the filtrate, right? So in here is the filtrate that comes from what we filtered out of the blood. So all that's in there. We can't let that go and be excreted in the urine. We would die because we need that stuff. Okay? So we absorb 70% of reabsorption occurs at the PCT, the proximal convoluted tubule. Okay? And again, we're talking sodium, potassium, bicarbonate, water, amino acids, glucose. So all these solutes that have come out of those crazy, wholly fenestrated capillaries of the glomerulus are now floating around in the filtrate, and we need to get those back in the body. So we're going to look at a piece of this, okay? So let's draw this and look closer to it, okay? And reabsorption is not like filtration in that the stuff just goes back up into the blood. It goes through, basically diffusion some, but it's facilitated, and secondary active transport also contributes to this. So it's a little different than just, filtration where it just comes out just because it's being pushed out. All right, so if we make our little cell, so here's our little tubule cell. Okay, so keep in mind up here, is the lumen. So this is where the filtrate is. All right. So down here is the basolateral area that's, stuck to the body, right? And so there's going to be a blood supply here, some capillaries. Okay, so these are, paratubular capillaries that are, around the tubules here. Okay, we talked about that. The paratubule capillaries are just all these capillaries that wrap around the proximal and distal convoluted tubule. Okay, so this is the lumen, so that means there's filtrate out there. And we need to get this stuff out of the filtrate and back to the blood. Okay, so this is basically facilitated diffusion, in which we use sodium to do this. So I'm gonna do glucose as the example of how this is done. All right, so we have glucose. We have glucose in the filtrate. It's been filtered out, right? So we got to get that back in. How much glucose should end up in the urine? 0. 0 glucose should end up in the urine. So we reabsorb all of it, okay? And we do this through the use of sodium. So sodium, we know, is out here. It came out too, and it's in the filtrate. All right. And, just realize too that, these are simple cuboidal cells. We should know that. Those are the cells that go, that are line the tubes, right? And you may not realize this, but these cells do have microvilli in this tube because we need to reabsorb 70% of what we just, filtrated out. So we need to be able to have as much surface area on the cell, the luminal side of the cell as possible. So for glucose, and this is for the other solutes too, we have a transporter, okay? And we know this transporter, it's called SGLT, or the sodium glucose transporter. So that transporter is here, and it uses sodium coming in to bring in the glucose. Okay? So again, we need the glucose to keep coming in. We need the sodium to keep coming in and cause this energy to bring the glucose in. So what do we need to do to maintain that? Well, it's the same thing that we did with the digestive system. There's going to be the sodium-potassium pump here. So that pump. So it will take the sodium and pump it out into the blood. Okay. It exchanges for, it's the sodium-potassium pump. So now we get potassium coming in. Okay. So we have another transporter on here. And it's one we've seen before. Good color here. And this transporter is Glut. So it's a Glut transporter. Again, we saw that before, and it brings the glucose out into the blood, out of the cell and into the blood. Okay. We know that potassium, because there's so many open channels for potassium, it just goes back in. Okay. So now we have glucose coming through here and going into the blood. We've got sodium coming in here going into the blood. What else goes with that? Well, if you're thinking water, then you are correct. So water is going to follow that solute right on through. So this is just osmosis going on here. So through osmosis, it's going to follow those solutes and go right into the blood. So in this just moving glucose, we're getting sodium back into the blood, we're getting glucose back into the blood, and we're reabsorbing some water here back into the blood. So now, because these solutes are leaving the filtrate, the osmolarity of the filtrate is decreasing. So it's becoming less what's the word? So it's not dilute, it's the opposite of diluted, okay? So, so we're getting, again, we have less solutes in there, okay? So now the osmolarity is decreasing and the filtrate is getting more dilute, okay? All right, so the concentration is changing there, okay? All right, so that's basically what happens here. Now, there is something we're going to talk about in the next video that goes along with this. All of these solutes travel basically this way, just like glucose with transporters. But here's the thing, the transporters are not limitless, okay? So we'll talk about that in the next video.